In this side view, the first cleavage or division begins from the top of the animal pole and slows down as it gets into the denser yolk material of the vegetal pole. From the top, we watch the first cleavage again as it divides the fertilized egg into equal halves. Second cleavage occurs at right angles to the first division, and now we have 4 equal cells. Third cleavage occurs across the 4 cells, around the middle, creating 4 more buoyant animal pole cells. We watch the next division from the side to emphasize the disproportionate symmetry caused by the dense yolk material. Back at the top, division keeps occurring, with the rate speeding up as the cells get smaller. Some cells are getting pushed toward the center and so appear to be smaller, but all divisions are still equal at this stage of development. The cells of the vegetal pole divide more slowly because of their density, so there are less cells in that region, but lots of cells at the top. Presumptive endoderm and mesoderm cells at a contact zone become active and move into the blastocele, starting gastrulation. While they continue to divide, the cells migrate inside, closing the blastopore and forming the yolk plug. To go from a sphere to a frog takes a big reorganization of cells. In gastrulation, outside cells invaginate into the inside of the sphere, setting up the future form of the body. Cells along the neural groove differentiate and grow, causing a swelling, then a folding, eventually forming the tube that will become the dorsal hollow nerve cord. One end grows and swells even more, giving rise to all parts of the brain, which now defines the developing head region and establishes bilateral symmetry.
The first cleavage divides the zygote equally in half, making 2 cells within the fertilization membrane. The second cleavage divides each of the 2 cells equally in half, making 4 cells within the fertilization membrane. The third cleavage occurs at right angles to the first 2 cleavage planes, and again divides all cells equally. As each cell's mass gets smaller, division is accomplished more rapidly. Since sea urchins have very little yolk, only a few cells at the vegetal pole are affected, and most of the cells show equal size divisions through the morula and then keep growing and dividing to form the hollow sphere of the blastula stage.
This plant nucleus is in late interphase. The DNA gathers into chromosomes and duplicates. In prophase, the nuclear membrane and nucleolus disappear. Chromosomes move to meet in the middle at metaphase. Centromeres divide, and chromosomes are pulled away to opposite poles in anaphase. Then the cell plate is laid down in telophase. This plant nucleus is in prophase. You can watch the nucleolus disappear while the chromosomes are forming. The nuclear membrane also disappears. The chromosomes move to meet in the middle, on the equator, at metaphase, and attach themselves to the spindle fibers at their centromeres. The centromeres divide, allowing daughter chromosomes to be pulled away from each other to opposite poles in anaphase. The cell plate forms down the middle, differentiating the daughter cells, and the chromosomes disappear in telophase.
The eyepiece is mounted on a swiveling body tube. The rotating nose piece holds the objective lenses and is used to change magnification. The coarse adjustment knob moves the stage for focusing, as also does the fine adjustment knob. With the shortest, lowest power lens in place, slide a slide into the slide clip. Using the slide mover, position the specimen over the light beam and bring the stage all the way up. This is the eyepiece, the body tube, the rotating nose piece with objective lenses. The stage, the slide mover has upper and lower knobs. The coarse adjustment moves the nose piece up and down for focusing, as also does the fine adjustment. The rotating nose piece allows you to change magnification. Always start with the shortest lens and work up in power. Under the stage is the light switch and the iris diaphragm. The diaphragm lever can open worlds to you. Some things are so thin that they're invisible until you close down the diaphragm. With the shortest lens in place, use the coarse adjustment to bring the slide and lens close together. Now focus. Rotate to the next power and focus. As you switch to the next higher power, watch that the lens doesn't hit the slide. Now, only focus with the fine adjustment. The space between the lens and the slide is the working distance. It decreases dramatically as the magnification power increases. Through the scope on scanning power, switching to low power, focus, switch to higher power, center and focus. Using the coarse adjustment, focus on the slide specimen using only your right eyepiece. Now, use the focusing ring on the left eyepiece to bring the specimen into focus. Move both eyepieces together so the images fuse into one. Air bubbles can look like protozoa until you focus through and see nothing. Living things are not flat. You have to constantly focus as you scan across the specimen. Each cell is a miniature system contained within a semi-permeable plasma membrane. As we pass within, the various organelles can be seen floating in the cytoplasm. We enter the nucleus through a pore and immediately see strands of DNA in a large dark nucleolus. Leaving the nucleus, we enter the ribosome-studded membranes of the rough endoplasmic reticulum, the site of protein synthesis. On the edge of the rough ER is the net-like smooth endoplasmic reticulum, which lacks ribosomes. Products produced in the ER are packaged for export in the Golgi apparatus. Here, a packet of material is pinched off from the Golgi. A lysosome is seen ingesting a foreign particle. A mitochondrion contains folded membranes which are the site of energy reactions such as glycolysis. Centrioles are involved in cell division. Together, organelles and membranes make up the complex structure within the cytoplasm of the cell. From the nucleus, a strand of messenger RNA passes into the cytoplasm. During protein synthesis, the ribosome moves along the messenger strand. Transfer RNA brings amino acids to the ribosome. Bonding occurs, and the protein chain grows.
Coming from the embryo in this monocot corn seed, the young root or radicle grows down. The coleoptile, which shields the tender shoot, grows up through the soil. The shoot breaks through the coleoptile, grows, and starts to photosynthesize food for continued growth. At the same time, lateral roots develop and increase the absorption area for the fast-growing plant. The heart can be attached through an electronic transducer to a recording machine to graph its activities. First, we need to observe and record the heart's normal beating rate and strength. Watch what happens when acetylcholine is put on the heart muscle. The heart still beats, but dramatically slower. The chart recorder counts the number of beats per minute for us. Compare these counts to the normal heart rate. Epinephrine, or adrenaline, is another neurotransmitter substance which affects heartbeat. You can see its effects are opposite to acetylcholine, since it speeds up the heartbeat. Other chemicals also affect the heart. Tobacco contains nicotine, which we will extract here with Ringer solution of normal body salts and water. When applied to the heart, nicotine at first causes a stronger and then more rapid heartbeat than normal. I bet you can guess what is going to happen now when we add caffeine to the heart. Yes, it speeds up the heartbeat rather rapidly.
Skeletal muscle needs a stimulus to contract. The nerve is draped over the electrode and a stimulus is given, producing an action potential. With the electrode on the muscle, we can demonstrate the graded strength principle. Up to a point, contractions get stronger as voltage is increased. Stimuli are recorded by the little upward blip on the time event marker. A muscle won't contract until it is given a strong enough stimulus, the liminal stimulus, after which it increases in strength until it reaches maximal and increases no more. Measure the contraction heights to determine when the maximal occurs. When two stimuli are delivered rapidly, the two contractions will fuse. If faster, they will summate. Multiple summations produce a stronger contraction. As the rate of stimulus is increased, there is less time for rest, but a sustained muscle movement is accomplished. A very rapid delivery of stimulus allows no rest at all. But the contraction gets stronger until all available energy is used up and the muscle fatigues. Muscles don't just move, they do work, pushing and lifting. In this experiment, we want to see how strong this muscle is. At each contraction, 10 grams of weight is being added. Although the muscle appearance seems unchanged, the recording shows otherwise. Adding weights to a muscle will change the length of contraction until it is overworked. When a burden gets too heavy, the muscle's sliding filaments will pull apart. At what point did overloading occur? An overworked, overtired muscle will twitch by itself. It fibrillates with no external stimulus.
The major area for exchange of gases, nutrients, and waste materials is the capillary bed at the end of the arterioles. The blood cells travel single file. Ciliated epithelial cells line tubes or surfaces which need to transport something. By placing red blood cells near the cilia, we can watch how they are moved. Pressure from compression opens valves. Contraction of heart muscle creates compression, which opens the valve exiting the contracting chamber. Electricity causes contraction. This ripe follicle first expels clear follicular fluid. Then the ripened egg passes into the cumulus, which will surround it and protect it during transport. The cumulus is sticky, which helps it get caught on the fimbria of the oviduct and moved by its cilia into the oviducts. The egg is stained here, an egg and cumulus being moved by the cilia. Cells with no cumulus are just spinning in place. Now inside the oviduct, the egg and its cumulus is moved along by ciliated epithelium and by peristalsis of smooth muscle.
Subduction occurs as the ocean floor dives under the drifting continent and sinks. Volcanoes form and earthquakes occur. Lava erupts and solidifies. Continents drift. In the Mesozoic era, dinosaurs could cross from one continent to another. Extinction occurred. Their fossilized remains were buried in the rocks and drifted with the continents for 130 million years. Mammals evolved in isolation on separate continents in the Cenozoic. Dinosaurs look uniform. Mammals are diverse.